Okay, well, I think we can go ahead and start. I'm not sure who wants to begin here. Maybe you, Elijah. It seems like you kind of had a longer agenda than the others.
Sure. All right, so sorry, let me. All right. So the first thing on the agenda was just talking about procedure for starting the sharing, and I believe at our last meeting we talked about that the lowest hanging fruit was definitely the Woods End / Cornell soil sampling protocol, and in particular total carbon. But you're also welcome to share the entire spreadsheet of analysis that you got, and if people are interested in other soil mineral characteristics or other soil characteristics, we can definitely use that data as well. So I wanted to just talk a little bit about Jay has actually already sent me some files, which is awesome. But I wanted to talk about a little about what we kind of need to get things into the database and get that database set up. So obviously, if you share your output spreadsheet from either Woods End or Cornell as an Excel file, that's fabulous. The key component there, as well as this kind of standardized spreadsheet from those lab services, is that within the field name column, or wherever, whatever identifier you're using for your sampling points, those identifiers need to match what you then give me in terms of geographic locations for your sampling points, and or fields that those observations pertain to. And I can think of a couple different ways for folks to get those geographic locations to me. If you already have GPS points logged in somewhere for those sampling points, you can send those to me. Most GPS programs will export those as a KML file. If you just send me the KML file, I can upload that to our system. If you want to connect them to broader polygons, so like a field or a pasture, then we'd need to do one of several things. Either you can, if you are using a GIS-like system and you already have shape files set up, you can send those to me and I can import those into the system. If you don't have shape files, you could send me, just if you have a map where you've drawn out those borders, like a Google map, and you've drawn the borders on them, you can send that to me and I can use that to try to hand draw the borders in the GIS system, but I can't promise 100% accuracy. I will do the best I can from your map, but I can't promise it'll be precise. The other option is I can create an app and share it with people who need it, where you could actually walk the border of those polygons you want to input into the system, and then record whatever field identifier and some other information about that pasture or field plot. And then that will go directly into the system. And I can either send that to you and you can do it yourself, or we can make some sort of arrangement for me to come visit and walk those for you. I think with those, so really the GIS database will have two components. It will have the field polygons and or individual sampling points for the soil sampling. And I'm thinking with those field polygons, it would be helpful to have some standardized data. So, current use, maybe a list of practices that you have done on those particular plots, compost, tea treatment, things like that, something, whatever management activities that you're currently doing. What type of, is it a crop? Is it pasture land? Is it some sort of lay rotation, what kind of activities are going on, and then perhaps historic use. So if this was, historically an old hayfield, or, historically a parking lot, or whatever it was, you can record that. So that gives some interesting background data. And then the other part will be the actual soil data from those lab services. And as long as the identifier on the lab services matches the identifier on the point or the polygon, the name of the sampling point or the name of the polygon that data refers to, I can then join those in the ArcGIS system. So now the data from the soil lab service is attached to that particular polygon or sampling point. Does that make sense?
Elijah, that sounds awesome.
And so I guess we don't have everybody on, but if people wanted to chime in and let me know, like, do you have shape files? And if you don't have shape files, what would be your preferred method of getting your sampling points and polygons into the system?
Elijah, I have a question or two in response. I've been using FarmOS as a repository for our data, and so drawn all sorts of polygons on our maps, and there's a lot of information related to land use history. I've got soil health test data and soil chemistry test data linked to various plots. Though I'm very reliant on people that are better with technology than myself. So I didn't know if you like kind of speak FarmOS, or maybe I can reach out to Mike Stenta and pull shape files that are helpful to you.
Right. We don't use FarmOS. I mean, I could probably hop on a Zoom with you and you could share screen, and I could probably help you navigate through it. But if you have someone who's actually using FarmOS and has a working knowledge of it, they might be able to help you faster than I can.
Sure. Yeah, I'll lean on Mike. He's the developer of the platform, and he's very helpful. Yeah.
Yeah.
And Elijah, just a side note, that he's in part of Open Team, and so that's, it's a easy,
Right.
it meets some goals there too.
Great. So yeah, and if you just want to export those shape files and send them to me, then what I can do is get those in shape for upload to the system. And if there's data that's missing or data that needs to be recoded, I can just check that with you before, putting that into the system.
Perfect. Wonderful.
So I can jump in and add a little bit. The shape file, that makes a lot of sense, Elijah. I think that's our goal, is to send you our working ArcGIS files so that you've got access to all of that. And then I think we just need to update our land use history into ArcGIS, and then that'll all be embedded. If I can share my screen just for a sec,
Sure.
I can show you what I sent Elijah and Shane. So here's our version of our polygons. These are the field IDs we've sampled using the Cornell Soil Health Test in 2016, and then 2 years later in 2018. There are about 70 different blocks, and that's exactly like the idea of keeping the same field IDs within the lab ID. We just sent these to Will Britton, and that's definitely the redundancy that we got to make sure that the field ID matches up with the lab ID, and that goes in our spreadsheet. So it's been exciting to start to see over the years, to watch trends. So if we look here, this is just 2016 and 2018, we can start to see change in active carbon. Some fields, according to this, actually went down, as you can see in the light purple, and some, of course, increase. It's only 2 years, so there's really not enough time probably to draw a significant trend, but it's interesting nonetheless. And here's a look at respiration. So that's just our kind of initial interest in work with ArcGIS. And I think this carbon group is really exciting for aggregating this data in a meaningful way. So I shared with Elijah and Shane the Excel data from Cornell Soil Health Test, and then we'll add to that our 2020 test results that we sent to Will. And we should be able to get all of this to you guys. Hopefully the GIS files before Christmas is our goal, Elijah, and then spend the winter with updating our 2020 work. So then we'd have 6 years worth of trends to look at.
Fabulous. Yeah. And thank you so much for sending that. I think, other than the 2020 data, which you mentioned, the only other thing I would need to get you guys into the system is those shape files for those fields, and just, again, making sure those field identifiers match up with the lab identifiers.
Absolutely. Cool.
I have a question, Elijah. How many farms then are you collecting this data for? Are all the groups represented?
So we have, I forget the actual number. I think there were 5 or 6 farms that signed on in this little group to share the data so far, and that can certainly grow, but this will be a good starting point. And I think a lot of this is to start the process of sharing and work out the kinks and work out the procedures. So, and again, it may move eventually to another platform, but our idea was to really start sharing now and start working on all the procedures and using it as a jumping off point for discussion. And then we can always, perfect a system later, but we've started to build, and we've started the process of getting in the habit of sharing data with each other and the procedures for sharing data with each other.
Yeah, just to jump in for a second, Duke Farms has expressed interest. They do still want to be an active partner, but they've had a delay with Rutgers in getting any data. And so I have a feeling that's why Tom has been MIA in this group, but he did express interest in wanting to participate as soon as they have something to contribute.
Great.
Good to hear.
So I guess my next question is, all of our soil testing is, really at the 6 to 8 inch zone. So as we look to do deeper sampling next year, what's the depth that we should try and universalize?
Right. Well, that was the next thing on my agenda, was starting to talk about kind of sampling and standardizing sampling. Again, I think for this process, what we have is great, and we can make it work, and we can try to adjust and standardize as much as possible, but also recognize that there might be slight variations in sampling protocol. But then again, that gives us a jumping off point to discuss, do we want to standardize? If we standardize, what is that standard?
Okay.
I know that for us, it's actually not a standard depth. And this is actually, Will can probably jump in here and explain more, but based on his guidance, we started sampling, and I'm hoping that we interpreted correctly, but we basically have been sampling the whole A horizon. Some of our fields, that's maybe 6 inches, and others it's 12, almost 14. So we basically go until we hit subsoil. I don't know actually, unfortunately, that we have measured that depth when we take it, which would be helpful if we're thinking of total carbon calculations. But again, that would be in each, that would, sometimes it's worse to take measurements and assume that is enough data to draw a conclusion than to just not just, just take the data you can work with. So I don't know, Will, what is your opinion here?
Well, there's an old saying that standardization is a devilish word. And, if we standardized at 6 and your effective farming soil layer, which is my philosophy, is 8 to 12, that's unfair to you in a sense of not representing your real agricultural plant relevant depth. But if we're going to compare apples with apples, like with carbon, we're going to have to agree that one depth was what was sampled. The way around it would be just to report effective carbon per relevant soil depth per acre. And I'll talk a little bit about that later. But to do that properly, we also have to get good field bulk density measurements. And, you've heard the simplification that parts per million times 2 is pounds per acre. Have you all heard that in soil testing?
Yeah.
It's a gross misrepresentation of the situation. And I just read an article this morning from Penn State about how we arrived at that and how extremely variable it really is. And nobody wants to rock the boat on this, but since we're really launching on such high quality work here, I think we've got to question all these assumptions. And if there was a way in a database to have like one column showing effective plow layer and the next column over just showing 0 to 6 inch layer, then at least we have a mechanism to standardize data. It will mean increasing amount of sampling. If I told you, Shane, to go back and resample only the top 4 inches, you'd have to do the whole thing again because you mixed your sample depth when you did it. And I think everybody's in the same situation. But I'm working with some farmers that they're working, 12 to 14 inches of soil. When we dig down, we see the plant roots are fully in that depth. So why would you sample less than that to represent soil quality for the plants that you're growing? And there's no simple answer for it.
Yeah, I know when we dig into our alfalfa fields that we've got alfalfa roots going down, 3 to 4 feet deep sometimes, or in our perennial Kernza fields that it's down farther than the backhoe can reach. So we know there's carbon down there.
So you actually dug a pit in the Kernza field with the backhoe?
Yeah. Our safety officer got nervous. Yeah. I mean, it probably went down 7 feet before we lost reach, and we could still see tiny little root hairs of the Kernza.
Wow.
And isn't Hudson Carbon doing 1 meter deep soil cores presently? Didn't they talk about that in the last meeting?
Yeah, they're going down to that depth, but they are separating out depths within that range.
I guess what we were trying to do is get some of the lowest hanging fruit, in a sense, practice sharing the data and getting it on a map at all, and including enough for the qualifications so we don't draw conclusions that we can't, and then start to hone in on some of the standardizations or ways of accounting for the differences that would make the data higher quality. We also have set up some, we're collecting not every year, but every like 5 to 10 years. So last year was the first, down to 1 meter in 16 of our fields, but that, that data we can treat a little on a different map.
Yeah, I agree with that. It's almost, yeah, to pick the low hanging fruit now and to exercise the idea of sharing. I think that's a radical idea. Let's start there.
Well, and it prompts these discussions that and gets the ball rolling and gives us a reason to want to standardize instead of, you know, talking about it in the abstract.
Absolutely. So I'm not sure when a good time is, but I know Will's got some slides too to show of a project we did here at the Farm Hub where we did a bunch of different sampling. So it just depends on how much time we have and what else we want to talk about, and then I'm sure Will could keep us interested.
Yes,
Elijah,
so-
did you want to have any more about any sort of standardization specifics for now, not necessarily like sampling design for 2021, but relevant to right now, or do you feel good?
I feel pretty good. Again, you know, I can follow up with folks as they share their data and make sure we have the information on how this previous data has been sampled. But I think this is a good jumping off point to start talking about 2021 sampling, and are there protocols we want to put into place for sampling protocol and sample handling too, which can also have a big impact on those laboratory tests, how you collect the samples, how you handle them, how they're stored before shipping, how they're shipped. All that can have an impact as well, as I'm sure Will could tell us.
Yeah, as I said, the devil's in the details.
Yeah.
It's a fun voyage that we're all on. I mean, I can share my stuff at any time. As Jay mentioned, we started another spatial variability project, which I believe we've already mentioned earlier. So we're trying to figure out if high rate sampling has an impact on the soil results. So we have some good preliminary data on that I can share today. And we'll be getting this into spreadsheets, and it'll be tricky, Jay, to get this into the ArcGIS maps because we've subdivided fields now. But
yeah.
We could probably average it for the fields so it doesn't make it too difficult.
Yeah, if you guys want to share that, I say let's start with that, and that actually may provide some good discussion points for sampling design that we can either continue today or carry over to the next meeting.
Okay, so I can just grab the share button and let's see. So here we are. The project title is comparing traditional versus high rate sampling for soil testing. And today I'm just focusing on soil carbon and a few other related traits, but we're looking at all the nutrients. We're looking at the soil health indexes. We're looking at ratios of nutrients to see in terms of forage quality, if we were growing forage, how these different machines work. But on the cover here is this device that we tested out. Jay's team had them come down and do all this sampling. I think it was back late August, wasn't it, Jay?
Yeah.
And this is quite an instrument. So what we're trying to do here, let me go to my next slide. Traditional versus high rate sampling methods. You're all pretty familiar with sampling, I'm sure. And here we show Jay in the middle there doing, I call it high quality, slow sampling. When you do your own individual core sampling like this, you are in full control of it. He's pushing it in the ground. He can pull it out here and see he's got 6 inches of soil and validate that. The difficulty of this approach is nobody takes many samples. As you know, I often ask farmers how many samples they really took, and they said, oh, it was, you know, I got tired and I took 4 samples in the field. And look, that's a very, very tiny amount of soil in that core. So the problem here is, how can you validate that you are accurately representing carbon or anything else with a small amount of samples? So the normal process is used for hand coring is 6 to 12 cores per field. And as I said, it's a high level of self-control, which I think is great. It's tedious and time consuming. You can really feel sore in your tendons and your wrist at the end of several hours of sampling. You will even know it the next day. And the other thing interesting about this semi-automatic approach that we're using, it's also a core sampler. So in the top left picture here, that's what the cores look like on this big rotating unit. Then that's spinning around and jabbing into the soil as fast as they can drive. So it is taking a slightly larger core. I forget what the diameter of that was. That was at least 1 inch, wasn't it, Joe?
Yeah, it was 1 inch.
Okay. So, and your hand core is probably a 3/4 inch.
Yeah, yeah, or maybe a tad bit bigger,
I
3/4.
mean, it sounds like that's not much difference, but from a point of view of sampling variability and quantity of soil, it actually ends up making quite a difference. So here's what the Falcon looks like for those of you who haven't seen it. On the top of the picture is the big rotating drum that's spinning constantly as it drives the probes into the soil, and this soil falls in here, and then it's going up into the emptying tray and sliding down into a turret. And here on the bottom is the turret. So you can go through a field and be grabbing samples and filling any number of boxes over the space of the field. And it's very convenient. You know, the box is full here in the top left, and you pop it out, and they are capable of GPS IDing where they are sampling, and I think we're going to try to do some more in that direction. But look at this, it takes a sample every 15 feet. You can increase that to 7.5 foot distance by adding another core onto this big drum. For some reason, they didn't offer that to us at the time, but that would require you to have more boxes and be emptying it more frequently. But approximately, it's taking 200 cores per field. And I'll compare that to going out there and taking 6 to 12. So again, the idea or hypothesis is, can we get a better sense of what we have across the field taking more samples? So we did 2 of their fields. One was a 57-acre field and the other was an 18-acre field. And we agreed to divide it in what are called strips labeled 1, 2, and 3, and then subplots labeled A, B, and C. So we sampled each of them individually. So we're measuring the change in soil quality going away from the roads in each of these cases, 1 to 2 to 3, and then going along the length of the field, assuming there may be some variability across the length of the field. And within each of those, we did a zigzag pattern with the big sampling machine to basically collect as many as was reasonable. So at the end of the day, everything is composited into all these individual samples. So we have a 3x3 grid times 3 for each field. Just look at the quantity of cores these are the 2 fields. So what we did is we measured, we knew the area of the field, and we had subdivided it into these blocks, but we measured the sampling distance in feet traveled, and we knew what the diameter of that drum is. So theoretically, this is how many samples were being drawn within each of those zones, which is amazing. So the Falcon machine in one field took 1,134 individual cores while traveling 3.2 miles. And if you divided it into zones here, there's about 20 samples per subzone. And the fields were a little bit different. In the smaller field, we must have made more turns or something because we got a slightly higher average. But again, compare this to only 96 samples if you sampled each of these at the sampling density that we mentioned for traditional sampling. So, you know, the drum roll here is surely the Falcon with so many more samples will give us a different result if variability is being overlooked in these relatively small number of hand core samples. Maybe just, Jay, you could briefly say, to make sure the study was highly comparative, you went out and hand core sampled literally close to where the machine had sampled.
Yeah, the next day I just followed the tire tracks of the Falcon with a four-wheeler and got out and did the hand coring.
So we can't say it was identical, but it was very, very close. And the tire track, you could even see the holes where the Falcon had penetrated the soil, right?
Right. So I tried to match the same depth by
Yeah.
replicating them.
So these are all important things doing a study. If you present papers like this at scientific conventions, they say, well, how closely did you control all this? And so on and so forth. So here's the total carbon for the whole farm. And I like to do these histograms. On the bottom is the percent total carbon. And these blue bars are the frequency, the numbers of samples that fell into that range. And this, to those of us in the lab, this is a really nice curve. It's called a normal distribution. In the sampling world, you want to see your curve fit this pretty well, where that's the average right here, and it drops off across the field. So the average carbon for both these large fields are about maybe close to 100 acres was 1.232%. And the deviation of it was 0.27, so that's over 20% deviation, even with that large amount of sampling. So, but from a distribution point of view, it was a really great data set. So how did we do with the carbon group? So this is a graph showing the distribution of three forms of measuring carbon: CO2 respiration, total organic matter by combustion, and total carbon. And the colors here are the Falcon versus the traditional sampler. And we threw the distribution plots on just to show you how close we're getting. So in all cases, with the Falcon, we got higher levels of respiration, but it spread out more than with the traditional sampling that had a lower average, but a tighter set of results. Organic matter, the Falcon was higher with the exact same distribution as with traditional sampling. And total carbon, they were very close, but the Falcon was, again, higher, higher average, that is. But the distributions were fairly good, and the traditional sampling had more uneven distribution because of you can look at the frequency plots there, the samples were more variable, appeared to be. So here's a summary across the farm, and I did this in tons of carbon per hectare, since that's like a world standard. And then our statistic program shows you all the possible interactions here. So these are the three strips in the field, 1, 2, and 3, going across here. So in this scenario, field 22 was much higher and significantly higher in carbon than field 8, 7, and 8. But you see here, and the strips behaved almost identical, depending, the trend across them was the same. Now, when we go down here, we're now testing the different strips by whether the Falcon and the traditional could see them, see the differences. Here, you'd like these lines to fall right on top of each other, and they didn't. The Falcon was still seeing more carbon. And here, it's the field and the type by the sampling. Consistently, the Falcon found more carbon. I mean, again, you go up the line here and over, it's not huge differences. So in field 22, look how tight the two samples were, 1.35 versus 1.42 in carbon. And here at field 8, it's spread out more. But look how strong these differences are when you summarize them all. So the variability of total carbon in tons per hectare was very small, going from 1 to 2 to 3. So there's not a lot of variation going in from the road, but the field differences was very large. And we all know that fields are different. That's almost a 5 ton per hectare difference in carbon, just jumping from one field to the next. But look at this, the Falcon got more carbon tons than the traditional in the same fields measured across these, both of them together. So one thing I tried to do is ask another question. What about, did the Falcon change the nature of the soil? So one question was, did it affect soil bulk density? And it did. The Falcon showed higher bulk density. This blue curve is shifted over from the traditional by a very small amount, which means it got more soil. But it was sampling the same depth, which means it compressed it a little. And on this side, the overwhelming difference, though, is the field. So field 22 was just much less dense than field 8. So with all these situations you have to distinguish, are we seeing variability caused by the different fields or by the methods? And here we're seeing a combination of both. The other thing is, I was worried that the Falcon being mechanized would damage soil aggregates. This is the water stable aggregates. And here we compared Falcon to traditional on the left, and they're virtually identical distribution, which is really good. That means that machine was not like smashing the samples more or doing anything physically disruptive, because it's moving through the field pretty fast. And we could not validate how well it was sampling, because you just can't do that. You'd have to stop the truck and get out and take a look at it. We did validate, though, that the big difference in aggregates was, again, between the two fields. We've now just sampled the entire, literally the entire farm, right, Jay? Just about every field.
Yeah. Yeah, we used the Falcon to sample the entire farm. That looks like that shape file I showed earlier.
Yeah.
And then we also sampled the entire farm again using a traditional method. We contracted with Caravel to sample with their four-wheeler hydraulic mounted probes.
So imagine putting this, we're going to redo this, all these graphs now to show all the differences between fields and sampling methods. So here, the preliminary conclusions are that using this intensive Falcon sampling, we were able to observe more organic matter and more total carbon on the farm. The group variability was not decreased or lessened by the Falcon. One of my hypotheses was right from the beginning that we'll see less variability with the Falcon because it's sampling more, but that wasn't the case, which is very interesting. But look at this, the Falcon, using Falcon testing, we found 1 ton per acre more carbon attributed just to it. And here's the numbers here. This is the total carbon. Here's the bulk density of the samples. We corrected it to the weight in tons per acre at 6 inches. We converted it to metric tons. And look at the differences. So in pounds per acre of carbon, if you sample with the Falcon, you had 1909, or essentially 1 ton more carbon from your test results. That has value in a carbon market, right? So how do we address that? It's going to be really interesting. The other thing is 1 ton of carbon per acre is equivalent to 2 tons of organic matter difference in measurement accuracy. 2 tons is more than the entire crop residues after you plow under corn stover, right? So that, from one point of view, that's huge. And so this has just fascinated me that we're going to need to do a little bit more study with differing tools to see what the source of the difference is. And we haven't yet discovered it. We have several theories, and Jay and I talk every week about what the difference is. I want to believe that we found more carbon because the Falcon just did a way better job of sampling, accounting for more variance across the field and getting more highs and more lows, but they averaged out higher. A more gloomy conclusion would be that the Falcon got more surface litter in the sample. And we did observe that. It's just punching down wherever it happens to land, right? And I think as I was watching, and we did this, I was on Zoom while they were doing the hard work in the field, but I could see the head would come around and there'd be a chunk of corn stover stuck into one of the probe points, right? And that would fall out. But what if that's affecting the results? We don't have any strong evidence that that's the case, but I'm still looking at every other aspect of the soil test to see if it is the case, because then if that's true, you really want to use a high rate sampler only on a fairly clean field. If you're going into a field with, like, imagine wheat stubble, and it's just punching down and picking up plant matter and so on, that could have a real impact. Having said that, in the lab here, and I'd like to do a presentation sometime just to show how we process soil, we try to eliminate all these factors by screening out the litter. It's not really soil, but it turns out litter has a huge size range from, a 1/4 inch to a 1/2 inch, all the way down to a millimeter. And if it's smaller than 2 millimeters, it's going to go through as sieves and be tested as soil. So that's another source of variability in testing how the lab is processing. So I think it'll be fun if we all just keep working this through and sharing our ideas. I'm not doing this to sell more Falcons or anything. I'd like to save people some of the hard work of sampling. I'm working with another group that's using auger drills to do really fast sampling, like in the Quick Carbon group. And we've discovered there's some real issues with controlling sampling, even with augers, because you think it's pulling all the soil up, but actually it's spinning and leaving some soil behind. These are all the factors that have to be considered. So that's all I have to share with you now. I'd be happy to take any discussion about the slides.
That's fascinating, Will. Thanks for sharing that. I've definitely had that experience with the auger bits for the Quick Carbon, and as they dull, and the end goes from chiseling away the soil to being rounded, I started realizing I was getting like half the sample weight each time. And I think a good chunk of it, and who knows which portion of the sample, was just getting, I think, smushed against the side of the
Yeah.
hole. And
Yeah, it really worries me.
yeah. So it is a slightly troubling thing, because that does make sampling easier and quicker.
Although, have you done a lot of it? Because I have a pretty big power drill with an auger, and my wrist is exhausted after taking like 30 or 40 auger samples. It's really exhausting.
Yeah, it's not that much less physically taxing. Yeah, I've done quite a bit.
So they call it easy, or USDA calls it the no sweat sampling method. And I tease them and say, have you guys ever actually done it?
Yeah. If you hit a rock, it'll spin your wrists around and whack you.
Yeah, you got to drop the drill.
Yeah. um,
So this, I call this fun stuff. These are fun details. But, you know, if we tell the world, and I had a scientist from University of Michigan call me the other day, and he said, you guys have to be prepared for serious validation when you're putting up carbon figures for tons per acre, tons per hectare. You have to be concerned about how the others of us are going to validate that.
yeah.
And so we do have to be careful and be very honest about differences that we're experiencing. And we might spend the next 3 years just working on differences. And to me, that's okay. It might not sell film scripts or anything, but at least we're working it through.
Will, one question I had, I know the sample sizes were wildly unequal, but did you do any statistical testing on the difference between the Falcon and traditional sampling for the same field?
Yeah, that's what essentially the whole study is. It's a complex set of statistical comparisons of all the test traits by the different methods and by the different strips and the different subplots. So yeah, I mean, it's like what we call a randomized complete block strip study based on statistics, and everything that I've reported as significant had to be statistically significant in order to call it that. So like
Okay,
that,
so I can't remember. There was statistical significance between the carbon measurement for the Falcon versus traditional for the same field.
yes,
Yes. Okay.
yes. And I'll make sure we all understand and appreciate that. Some people glaze over when they see the statistical plots. And we use little asterisks to signify the different levels of significance. The good news is on that study, the differences between the fields carry the overwhelming statistical significance. That's where most the difference is. So the difference between the Falcon and traditional is certainly much smaller, but still it was greater than 95% probability in all the cases.
That's really cool to see. Well, I think that's the first time I've seen that that slideshow from beginning to end. So if I was going to want to go on a vacation with my carbon credits, I'd probably use the Falcon.
It's interesting thought, yeah. Obviously, we have to deal with sampling depths now. And nobody builds an automatic sampler that can go deeper than 8, I think. So it's back to hard handwork or using a hydraulic sampler, and those are very slow. I mean, for those of you who have used it, isn't it really slow to do a 1 meter depth? You get the thing planted in place and you slowly drive the core down. You can't do it fast, right?
Yeah,
Well-
we can't use a corer at all. We have too rocky of soils, so it's all
rocky.
by hand.
So it's really slow for us, hence the every 5 to 10 years. Maybe a backhoe we could use, but we didn't really want to dig 48 backhoe pits in our field.
Yeah, it's funny. I took a soil science class, and the class that I took, I actually had to dig a 3-foot pit in my backyard, which is pure rock. And so I feel you. Yeah, definitely. And I'm sorry to interrupt, but I just want to say we have about 10 minutes. I'm happy to go longer than that if you guys have time. But if not, it's about, you have about 10 more minutes. And I don't know if you want to go ahead and sort of figure out next steps or figure out the next meeting. I actually also wanted to ask you some questions about the website and getting your information onto it. So let me know, and then we can go from there.
Great. I just got asked to hop into another meeting at 2, so we can go a little over, but I'd like to not go too much over. So yeah, I think if we can take steps to wrap up in the next 10, that would be great.
Okay, cool.
Cool. I guess just in that spirit, Elijah, if you could email out a document of those, of the 4 things I think they were, of how you would like to see files shared, from everything from like, it needs to have the field ID, it needs to have the land use history, etc. I think that would be good for the group.
Absolutely. I'll do that. I think just one thing for my schedule to note is we have the Young Farmers and Chefs Conference coming up this week. So up until like Thursday next week, my bandwidth may be fairly limited, but after that point, it should open up.
Cool. Do you want to set up a next meeting for everybody, or do you want to do that
Sure.
email?
Should we, let's see. It seems like probably logical to go, maybe with the holidays coming up and stuff, maybe go a month and shoot for early January. Does that work for folks?
Yeah, it works for me. Yeah.
Scheduled Thursdays are generally good for Ned in case he wants to be a part of the next one.
Okay.
So January 7th looks pretty good right now. I don't know how that looks for you guys.
I think January 7th, as long as it's in the afternoon, is good for me.
Likewise.
Yeah,
I
2 p.m. to
think,
5
Jay,
is
we
open.
need at least a month to put all our data together.
Well, we're
We're
going to
try
need
to get
all
it into
winter.
the
All winter,
ArcGIS
Will.
map, so we're figuring out all the algorithms to upload from CSV sheets, and it gets complicated. Elijah, you could probably help. And we're doing a massive comparison of all the prior Cornell soil health traits with the Woods End one, so that'll be like a correlation study, because I think a lot of people are really interested in that. Because each lab has a different set of measurements. I just did a total carbon study with 4 different labs on the farm, Caney Fork Farm, and we're finding that the labs get slightly different total carbon results too. So that's another variable. So depending, the word is out that there's certain labs getting higher carbon, and so you could send your samples there and get more carbon credits.
You use the Falcon and then you send the samples to that
Exactly.
high carbon
We'll
lab.
optimize this for
Yeah.
everybody.
Yeah, and actually, Will, I'll say GIS actually would be, if the Falcon is getting the GPS coordinates on those samples, ArcGIS is really great at, you could feed me polygons and you can feed me those Falcon data points with the GPS attached, and ArcGIS will automatically summarize within each polygon. You can average or do any calculation you want on the data points
So
within.
that's great. We'll have to
the
figure
polygon.
that out.
Yeah.
Because Jay, didn't they explain as it rolls along, it's not collecting it for each sample point, but for the trajectory of travel,
Yeah.
something like that.
Yeah. It's got like a scent trail.
Which they are plugging into Precision Ag software by another package. That's my understanding. So
Yeah,
they
we
are,
need to figure that out a little bit more.
there's a lot of technical database type, just standardizing people's data streams is the big effort here. The worst thing is to compare apples and oranges.
But as far as Heather's question, I think so. Should we say 1 p.m. on January 7th as tentative time for our next meeting?
One,
You
not
need a
quite.
little later, Dave?
Yeah, I do. Sorry, I was just typing that as you asked.
Yeah. Let's do 2.
Yeah, I'm
then.
in a meeting and 2 o'clock will be better.
Okay, let's do 2.
Okay.
So that's 2 p.m. January 7th.
Thank you.
Correct.
And I will email all of my website questions just to save us time. But if you guys wouldn't mind responding to me, I would really appreciate it.
Great. And likewise, I will send out an email with all the steps for getting the data into the system and getting the field boundaries or pasture boundaries into the system. And you can send me questions or whatever you need to send me.
Sounds great.
Thanks. Exciting work, everyone. Good to see
Yeah,
you.
this is really cool. Good to see you all.
Take care. Happy holidays.
Bye-bye.
Happy holidays.
You all.